Hello, young anatomists. Let's create some drawing notes about the urinary system. Hopefully you've got a piece of paper like this from our class just to kind of help you plan some things out. I'm going to zoom in a little bit and start with just like an overview of the main functions of the urinary system. I can't think of a better color to use than yellow here. So the urinary system is going to be kind of its primary function is that it removes wastes. And I want to be a little bit more specific than that. And so when our body uses proteins, our body breaks down those proteins, and a resulting waste product of using proteins is ammonia. And ammonia is really toxic to our body. So then our body converts that ammonia to urea, and that is our nitrogenous waste in our body. And so the urinary system is going to be getting rid of that. And that's why urine is called urine, because it has to do with urea. And then number two, a main function of our urinary system is to maintain homeostasis for water and electrolytes. So water and electrolyte homeostasis. And how we're going to approach this is we're going to look at a little bit of gross anatomy in this box. Gross anatomy. So kind of like the organ system level of the urinary system. Then we're going to move to more of like the organs level down here. And then along the bottom, we'll talk a little bit more about the physiology and the process of urine making. Now, in the urinary system, probably the organ that's most well known is the kidneys, and the kidneys are shaped like little kidney beans. So let's draw a couple of those here. There's one kidney, and we have two kidneys. So those are paired structures, kind of like on either side of your back. So here, let's label it kidney. And another main structure of the urinary system is the bladder. So let's draw the bladder down here. And you have one bladder that's called the bladder or the urinary bladder. And the kidney is connected to the bladder by a couple of tubes. And those tubes are called ureters. Ureters, imagine it being like about the thickness of a spaghetti. And then, so urine is made in the kidneys, gets to the bladder through the ureters, and then from the bladder to the outside of the body through another tube called the urethra. And how urine happens is just filtering from the blood. Now, we've talked about the blood system to get to the kidneys in our circulatory system. Remember, there was like an aorta, the descending aorta that came down. And that's a big old honking blood vessel there. So let's label that. That's our descending aorta. And remember, we go to the iliac arteries here. So we'll draw those in. And next, how the arterial blood gets to the kidneys is through the renal artery. So we'll draw both of those branches. Renal artery right there. And then the renal artery kind of like breaks the kidney into sections when the renal artery gets there. So let's break that into sections there, and kind of like goes around the kidney like this. And then that's where the filtration happens in the kidney, the filtration of blood and separating out of the urine. And then let's draw our veins going back to the heart. All right, so there'll be veins in here in the kidneys parallel to the arterial system. And these veins coming back are the renal veins. That's really nice name there. And then going back up to the heart is our inferior vena cava, so IVC. So this is kind of like the basics of that organ system, some of the things that we have to keep straight here, and some good ties into our previous units. Let's scooch on down and take a closer look at the kidney. So here is, since I've got the blue pen handy, here is our inferior vena cava, taking blood back up to the heart, and then that branches out to the various sections of kidney once you're in there. And what brings blood from the heart is the aorta, and likewise, so I guess here, this is our renal artery. Should label that. That's the branch off of the aorta that then takes blood throughout the kidney to be filtered to get that nitrogenous waste out. Then that exchange kind of happens there, and then there's our renal vein taking blood back to the heart. Now there's a couple of sections that I want us to kind of like keep track of here. I'm gonna use a black pen for this. So this outer section of the kidney, right about there, that's called the renal for kidney, cortex. Cortex is a word that we see a lot, meaning kind of like the outer layer. And then the inner layer of something that we've seen before is called the medulla. So where do I want to write this? I'm going to squeeze it in down here. Renal medulla, like the inner part of the kidneys. And now a big part of the kidneys, like the functional unit of the kidneys, they say, is called a nephron. And so we're gonna draw a nephron in here. And how a nephron works is there's a part that's kind of like up here that we'll get into. It's what's over here. But a nephron starts up here. It has a big loop that goes down, and then that loop comes back up. And so this guy, that's called a nephron. And there's about 1 million nephrons, 1 million per kidney. And the nephrons are going to be doing all of the dirty work. And so what I want to do now is zoom in on one of those nephrons right over here. So let's scooch on over. A nephron has a cool little part here that's called the, like together it's called the glomerular capsule, if I can say that. And this is the part of the kidney that interacts with the blood. So let's go ahead and draw some blood vessels in. So we're going to draw an arteriole coming in, and then we're going to draw a bunch of capillaries in here. This capillary bed is called a glomerulus, and it's from a Latin root that means ball of yarn, and hopefully you can see how it gets that name. And it's really nice to have all of these capillaries here to increase that surface area and the chance for filtering blood out into the kidneys, into a nephron. This arteriole is called the afferent arteriole. And then this arteriole is called the efferent arteriole. And what's really weird about this is that, normally when we think about arterioles and then an exchange that happens, the arterioles go to capillaries and then turn into venules and turn back into veins. And in this case, we're doing arterioles for both. And so what that means is that blood is still moving away from the heart. Afferent means going towards the glomerulus, and efferent means moving away from that glomerulus. So let's talk a little bit about the specializations or form follows function of the glomerulus, knowing that we're trying to filter things out of the blood. So our glomerulus, what's that all about? The glomerulus is made of a lot of capillaries, and that's really important. That, like increases the surface area, increases the opportunity to exchange materials. Those capillaries are particularly leaky, so that makes it so that a lot of things can get out of there. The reason that these capillaries are leaky is because it's called fenestrated, and fenestrated or fenestrations, those are like holes. So let's say we're gonna call it holy. These are some holy capillaries, and everything passes out of those capillaries except cells. So these fenestrations, they're really big, and the only thing that doesn't pass through, so these only trap cells, like red blood cells, don't pass through there, but everything else passes through. And another cool thing that's there, and I would want you to look this up in your book, that there are cells called podocytes. And those podocytes cover the capillaries. Ooh, we are running out of room down here. Podocytes cover capillaries, and they have gaps, the podocytes, to let everything through, but keep proteins in. So a lot of stuff is coming out here. Let's draw that in. I'm gonna use a green marker for this. So here's all of those materials leaving the glomerular capillaries. So remember, the fenestrations are keeping cells from moving out, and those podocytes are keeping proteins from moving out. That process of moving materials first from the blood into the nephrons is called filtration. And we're gonna write the steps of urine formation down here in each one of those squares. So what's filtration all about? That's when stuff leaves the blood and goes into the nephron. And we'll talk about what that stuff is. I mean, really, it's everything except for cells and proteins, right? So let's talk about what's that stuff. Let's make this list. So that's electrolytes. And we said that the urinary system is in electrolyte homeostasis. So that's important for us to keep track of, and water homeostasis. So let's put water on that list. Water's moving out. Glucose, wastes, amino acids. And what moves this out is that this is driven by blood pressure. So it's important that this is arterial blood that's coming through, so that's high pressure blood squeezing against the wall of these glomerular capillaries and pushing everything out. All right, let's talk about those fenestrations here. I am off of the camera. Fenestrations. And so what are those? Those are holes on glomerular capillaries. So holes on the glomerulus that let everything but the cells pass. And then our other specialty are these podocyte cells. And what's so special about those podocyte cells here? They let everything but the proteins pass through. Everything but the proteins pass through. And when you, the amount of filtration that a normal person has, so the liquid or the stuff, the solution that comes out is called filtrate. And there's about 180 liters of filtrate per day. And if that's like 4 liters per gallon, roughly, that's a lot. I'm trying to do that math in my head. 50 minus 5, That's like 45 gallons of pee, and nobody pees 45 gallons a day. And so we're gonna like reclaim some of that back in this process. And there's an analogy of how urine formation works, and it's kind of like when you have to clean out your refrigerator. So to clean out your refrigerator, you take everything out of the fridge, and then you put back the stuff that's good, and you throw out the bad stuff. So let's write that analogy here. So this is like emptying the whole fridge. The only thing that doesn't come out, again, is those cells and those proteins, thanks to the structures that are in place. Now, the next part, this is going to be reabsorption. Reabsorption. Spelling that right? Yep, got it. Okay, reabsorption, and what we're doing there is bringing back what the body needs. So that's putting that good stuff back in the fridge. Now, this is going to happen in a couple of places of the nephron. So let's kind of like set that up down here, and then we're going to go back up here and like label these parts of the nephron. So about 65% of reabsorption happens in a spot called the proximal convoluted tubule. We'll label that up in just a second. I'm going to leave a little bit of space. About 15% happens in the nephron loop. All right, so adding that up, keeping track of the math, that's 80%. Another 15% in the distal convoluted tubule. And then the last 5% in the collecting duct. All right, let's go figure out what all of those parts are up here in our nephron. So scooch on back over. The good news is that as difficult maybe as some of those terms are, it gives us some good information. So the proximal convoluted tubule is right here. When you look at the picture in your book, you're gonna see that that this tube is like really wavy and really kind of like folds back on itself. So proximal convoluted tubule, PCT. Proximal means closest, right? And so we're talking about closest to the glomerulus here. Convoluted means it's all twisty and turny. And just like our small intestines are twisty and turny, that gives things like a lot more time to pass through. And tubule, well, because it's a tubule, right? So PCT. In terms of like anatomy and stuff, this part right here of the nephron, that's really important because it kind of forms like a cup around the glomerular capillaries. That's called the glomerular capsule. So that's really nice that it forms a cup around that glomerulus to catch any filtrate that's coming through. All right, let's talk about this. Let's finish labeling all of this nephron. And so this part right here, that's our nephron loop down here. And the nephron loop, so I'll label it, nephron loop. The nephron loop has two parts that have like really nice names that are easy to remember. This one is called the descending, because it's going down, the descending limb of the nephron loop. And then we're scooching back up here. And then this one's called the ascending limb of the nephron loop. And then hopefully you've kind of guessed it if you're going back here and where this stuff is happening. So then up here, this is our DCT, distal, because it's farther away. Convoluted, because it's all twisted up. Tubule, because it's a little tube. DCT, distal convoluted tubule. And then all of this stuff moves. So glomerular capsule, PCT, descending nephron loop, ascending nephron loop, DCT right here, and then finally into the collecting duct. All right, so if we drew kind of like some arrows to show directionality here. We've already pulled our filtrate out. We're going to do some reabsorption as we move through the PCT, the nephron loop, the DCT, and then we said there's 1 million nephrons, and they all go into this collecting duct, and then that collecting duct is going to go to the ureter, and we'll get to that in just a second. All right, let's talk about some specializations of the proximal convoluted tubule, because that's really big. This is a cool form follows function. So those convolutions, or twists and turns, convolutions do a lot of things. So that will increase the surface area to be able to do reabsorption. And that also, all of those twists and turns increase the time for reabsorption. So there's more time. Oof, I'm not gonna fit that in there. There we go. There's more time and there's more ability there. One of the cool things about the proximal convoluted tubule is that it has an epithelial lining that has some cool cells on it. So if we drew some of those cells, we would draw them like this. And then we put these structures on there. And as soon as you get these twists and turns anywhere, the goal is going to be to increase surface area. So we saw that in the small intestine to increase the surface area for absorption. And we're doing the same thing here. So that's a cool thing about the body as well, that it uses the same structures to accomplish the same jobs. And so these are called microvilli, or in this case, some folks call this the brush border. And those microvilli, or that brush border, have the function of increasing surface area to increase the efficiency of absorption. So let's talk about exactly what's being reabsorbed, because different things are being reabsorbed at different times. So in the proximal convoluted tubule, we want to snatch back that glucose, our blood sugar, because that's going to be really important for making ATP. We're going to be bringing back some amino acids. Maybe I should have written that whole thing out. Write it all out if you're not sure what AA means. We're going to be bringing out some sodium, and bringing out some chloride ions, and bringing out some water. And the saltiness of this it's a really genius thing, but it's hard for us to have time to really manage that in our course. So I want to draw some things coming out. In this part, sodium coming out here. Glucose coming out. Chloride, so those are electrolytes. Electrolytes are ions. Amino acids, out here I'll write it out instead of writing AA on there. Amino acids, and then water, all coming out. Next, talked about 15% of this process happening in the nephron loop. Main things coming out there are water and sodium. H2O and Na, same thing down here. And then in the collecting duct, we're pulling out some water. So let's draw that in here. In our nephron loop, we're making this area really salty by pulling out some sodium. And then once the sodium is pulled out, water will follow. All right, distal convoluted tubule, we've got water and sodium coming out. Let's draw that. I'm using blue here just because we'll add some more arrows for the other processes that are going to be happening. Sodium and water, and then finally in our collecting duct, pulling back some water into the body over there. All right, so if you think about what's happening in this reabsorption process, something that helps me think about this process is kind of generalizing of what's happening. So in this part, I like to think about this as the reclaiming process. These are like the really valuable things that the body wants back. And then by pulling out all of this water and sodium, this is really concentrating the urine, And then last of all, this is just like fine tuning. Do we need to get rid of more water or do we need to pull in more water? That's what's gonna be the fine tuning in the collecting duct. So in the analogy of our fridge, first you empty the whole fridge and then you're gonna be put most of it back in the fridge. So when you clean out your fridge, not everything needs to go in the garbage, but like 99% of stuff is gonna go back in. Let's move to the next step. That third step is called secretion. And in this analogy of emptying out the fridge, like after you do all of that stuff, our guy in Crash Course said, this is like emptying your pockets. So any like last minute wastes that need to go back in. So let's kind of like summarize that. This is the last chance for moving substances from the blood to the nephron. And if we think about like where this is happening, so a lot of it's going to happen in the distal convoluted tubule, but some is going to happen in the proximal convoluted tubule as well. What type of things do we want to make sure that we get out? We want to make sure that we get out any urea. Another form of nitrogen waste that we talked about up at the top is ammonia. So these two waste products. And then this is where like drug metabolites could go into the urine as well. Like if there's a drug P test, this is where those metabolites come in. Or, I mean, like it doesn't have to be like illegal drugs. It could be from taking like Advil, and the metabolites would go into your urine there as well. So this is going in the PCT, proximal convoluted tubule, and this is going on in the distal convoluted tubule as well. Let's use some orange to put these structures back in. It's a little tight here, and those are some big words. So instead of writing the words, I'm just gonna draw some orange arrows to remind us that here's where the tubular secretion is happening, and then also in the distal convoluted tubule. So a couple of arrows right there as well. And then the last part of urine formation, let's use this color right here, is called elimination. Elimination of urine. All right, so what's that all about? Well, each of those nephrons, so many nephrons, will empty into each collecting duct, Right, so that's what's happening right up here, going into the collecting duct. And then there are many collecting ducts. Emptying into each ureter. So there's going to be like nephrons attaching up here, and nephron, and then all of these are doing the elimination of urine, going to the ureter. All right, and then about only 1% of the filtrate that was taken out in the first place becomes urine. So if we go back and we think about that, right, 180 liters of filtrate per day, So 180 liters per day of filtrate. And that gets, by the time we're at urine, that's about, so what we said, 1%. So 18 liters per day. No, that's, sorry, 1% of that is 1.8 liters per day of the urine. 1.8 liters per day. Sorry, I really botched that. So that's about a half of a gallon per day, or about 8 cups per day. Just to let you know, like, what should be about normal, so you can keep track of how things are going in your body. All right, we've got a couple more things here. So I want to just kind of like trace that pathway again, and just kind of like put things in perspective as one long process. And then we're going to do a homeostatic loop. So let's trace what happened here. So our process started in the blood, right? So I'm going to use yellow. All right, so blood, and in the glomerulus. So we're just taking the whole path here, and I want you to visualize where we're at in your picture as well. And then next, that fluid went into the glomerular capsule. Glomerular capsule. So we're up here, moving right along. We went to the proximal convoluted tubule, then the descending limb, and the ascending limb of the nephron loop, or loop of Henle is what it's called again, but those names, that's after a person, and a lot of those people names are just going away in favor of things that are like more descriptive and good for the physiology aspect of things. All right, distal convoluted tubule, then collecting duct. I have to wrap this around. Collecting duct, and then collecting duct to the ureter. And that's where we left off on this picture. And then now we can kind of move over here. The ureters empty into the bladder. And then from there, the urethra, and then out into the toilet or wherever this urine is going to go. What do we call this fluid at these different steps along the way? Well, at this point, that fluid, let's use black. So at this point, that fluid is going to be blood. Once that blood passes into the nephrons, now it's called filtrate. And then after this reabsorption happens, that's where the filtrate really kind of like turns into urine. So somewhere in this spot, I'd say we could call that urine. And then that urine passes through and is eliminated in this process. All right, the very last thing that I want to talk about is water homeostasis. So let's put a little title up here. For water homeostasis. And we're going to use our favorite little organizer here for how this works. So remember, our 6 parts for every homeostatic loop is our set point and our stimulus and our receptor, our control center, our effector, and our response. And so our set point, like there's not really a specific number that we have to keep track of, right? So we're just gonna call this set point water balance. Water balance, like your normal water levels. And what we're keeping track of is if there's a stimulus of decreased water in your blood. So then we know that our response, what our body is going to do is make changes to increase the water in the blood. And I hope in your brain, like some things are starting to connect, like, okay, I see how we can do that here based on the mechanisms at play. The receptors for this are called osmoreceptors. If you know about osmosis, that has to do with water, and that can help with this root of osmo right here. So there's like water receptors in the hypothalamus. That's really nice because we've seen hypothalamus with homeostasis all throughout the year. Moving on to the control center. All right, now we're going to switch gears. The control center is the pituitary gland. And if a correction is needed, the pituitary gland is going to signal to the effectors. Pituitary gland makes ADH. So that is a hormone, ADH. ADH stands for anti-diuretic hormone. A diuretic is a medicine that makes you go pee. Anti-diuretic means it's gonna make you go pee less because we need to keep some water. All right, next, the effector. I should have made more space for myself there. So our effector is the kidney, like obviously, but more specifically, the distal convoluted tubule and the collecting ducts reabsorb more water. And then that is going to increase water in the blood and then bring us back to water balance. Now, I want to make a quick connection here. So the DCT and collecting ducts reabsorb more water. Look way back over here. Remember, after we do the reclamation, then we're concentrating the urine and fine tuning. So right here, how much water is pulled back, that's going to be where the anti-diuretic hormone comes into play. So let's draw some arrows in there just to kind of like help us make connections here, back to here. So if there's anti-diuretic hormone, now we're not just going to pull back like one arrow's worth of water. We're going to pull back more water. So that's more H2O in response to signals from the anti-diuretic hormone. And then the same thing here. We're not just going to pull back a little bit of water, but when there's anti-diuretic hormone because blood water levels are too low, that anti-diuretic hormone is going to pull back that water. I don't know about you, but I like at the end of these, like I appreciate all of this information in one spot, and then I like to just pull back and appreciate like what we did here in this process. So let's take a look after I get my finger off of the camera. That is a good looking set of drawing notes about the urinary system. And we can always find more details in our book, but this is going to be a huge study tool for us.